Hi everyone, it's a pleasure to be here today. So I'm going to talk to you about how we can use DNA language models to look and study nucleotide dependencies in the genome and eventually arrive at maps like this here, which showcase the dependencies for a specific sequence. So DNA language models are trained only on genomes to reconstruct a nucleotide given a specific sequence context by assigning a probability to each one of A, C, G, or T for the specific position. And here you see an example of a region in the genome of yeast which contains a tRNA. And you see the reconstruction per nucleotide and position. And you see that within the tRNA region, this reconstruction is much higher, presumably because the identity of the nucleotides is more constrained. And this highlights something that we and others have seen, which is that DNA language models tend to reconstruct functional elements in the genome. But we go one step further and we ask, given a query nucleotide, how can it affect the reconstruction of a target nucleotide? So here, back to the example of the tRNA, you might know that the tRNA also forms a specific secondary structure. If you take a query and the target, which are in the extremes of this tRNA sequence, but actually bind within the tRNA, the exercise we do is the following. So we given the query nucleotide, we mutate it to each one of the three other possible nucleotides, in this case A, C, or G, and we check what happens to the reconstruction on the target nucleotide in terms of odds ratios. And you see in this example, if you mutate the T to an A, then the model predicts with a higher likelihood the T in the target, presumably because it wants to compensate the target to be able to still bind to the query. And if you change, for example, to the G, then the model predicts a higher likelihood for C and for T, because in RNA you also have this wobble-based pairing that the G can bind with the U. So for a value, we extract a value for dependency between query and target, which is the maximum value within this table. So we are interested in how can a query maximally affect the reconstruction of a target. And on a sequence of n nucleotides, you can have about n squared dependencies. So we compute these dependencies for a specific sequence, and we plot it in the shape of a map, where each row is a query nucleotide, and each column is a target nucleotide. So given back from the example we have seen before, this would be the query nucleotide, this T, and here in the columns would be the target nucleotide, the A. And the value we take is the maximum on that table we've seen before. So one of the patterns that really pops up of higher dependencies are these anti-diagonal patterns, and they actually belong to each one of the arms of the tRNA. But we know that the tRNA also falls into a specific tertiary structure. And we also see some contacts that belong to the tertiary contacts, which help defining the classical L shape of the tRNA. So let's step back and see what we have achieved. So actually, we are able to get functional interactions from a language model which didn't focus specifically on tRNA loci, without any kind of sequence alignment and without any kind of biophysical rules. So we compute these maps genome-wide with different DNA and RNA language models, and we find different patterns. So we find local dependencies like on diagonal blocks, and we also find some distal dependencies like we have seen with the tRNA, some anti-parallel dependencies, parallel dependencies, asymmetries, and off-diagonal blocks. So I'm going to characterize a few patterns and tell you how you can use to achieve certain analysis and insights. So let's look here at an example of a promoter in yeast. You can see the reconstruction from the language model on each nucleotide and its position, and you see that mostly the reconstruction is highlighting transcription factor binding motifs, and is also highlighting this poly-D-ADT rich region. If you look at the dependency map, you check that actually the transcription factor motifs appear as blocks. That is because in the motif, each nucleotide depends on each other. And this is in contrast with regions like this poly-D-ADT. So from the maps, you are able to distinguish between highly reconstructed regions that are, for example, motifs or other lower complexity elements or other kinds of elements. So we derive a score that tells you how blocky each position is along the main diagonal, and we use it to, without any fine-tuning on experimental data, predict whether a nucleotide is bound by a transcription factor or not. And we do this across yeast, and we find that this score is as good as the PWM, which has been obtained by experimentally created data at predicting whether a nucleotide is bound or not by a transcription factor. And notably, just taking the conservation score will not give you any meaningful information, and taking only the reconstruction from the language model is not also does not also achieve a very good result. So we not only see blocks along the diagonal, but we also see blocks that are off diagonal, and they usually belong to motif interactions. So we take here an example of splicing. So in splicing, you have your intern, which is in the boundaries have the donor and the acceptor, and there is also a branch point. And you see in this case the donor depending on the branch point and the acceptor. And in fact, here in this case in yeast, interaction or dependencies between donor and acceptor are higher than dependencies between donor and decoy acceptors, so sequences that look like an acceptor but are not the canonical acceptor, or dependencies that match distances. In human, we also see dependencies between the donor and acceptor. This is an example taken from, computed from the model SpliceBird, trained on RNAs across different vertebrates. And you see here an example of an individual in GTEx. So this is a sashimi plot. An individual on GTEx which has a variant on a donor. And this variant induces exon elongation, as you can see here. And the map indicates, so when the donor is a query, that you have here the alternative donor popping up. So it is indicating that if you destroy the donor, if you mutate it to different nucleotide, another alternative donor changes in likelihood, and this alternative donor position belongs exactly to what we had seen in the case of this individual. So we then look at how dependencies between variants and their corresponding outlier junctions in human, in GTEx. And we see that across different locations for that variant with respect to the exon and intern, you have a higher dependency than at matched distances. So as you had seen before, we also see some anti-parallel dependencies, as in the case of the tRNA. And the good thing about these maps is that you can treat it as an image and use image processing tools to search for specific patterns. And in this case, you can use a very simple convolutional filter to look for anti-diagonal, anti-parallel patterns in these matrices. And indeed, the top patterns, the top hits that we see belong to RNA secondary structures, such as in this case, and not only belong to secondary structure, to the stems, but actually, for example, to pseudonodes, these non-nested contacts that are hard to detect computationally. So actually, if you take these maps and you take it to predict the contacts from structure directly without any fine-tuning, you can already achieve a very good performance. So this prompted us to actually search for anti-diagonal patterns that are not mapped to any known structures, and we did this in E. coli. And we identified different maps in different positions in the genome, which according to the dependency maps could be structured regions. And in collaboration with Dani Incarnato, we have validated several of them, and this is just an example of a novel structure where you see and the assay applied here was DMS map-seq, which measures the reactivity of a nucleotide to DMS. And the more reactive the nucleotide is, the more likely it is in a non-structured region. So you can see here that most reactive nucleotides are on this non-structured region. And the nucleotides that are in the structure, you can see that they belong to stems, which you can map to the dependency map. And also the dependency map is highlighting here a pseudonode, which you can see this stretch of views, which are indicated to depend on this stretch of A's. so indicating that we are in the presence of a pseudoknot structure. Okay, we also see other kinds of patterns like parallel dependencies. And here is an example of the reconstruction here of a language model in a sequence where you have an element and its repeat right afterwards. And when you have this, the first element of the repeat depends on the first element of the other repeat. The second element of one repeat depends on the second element of the other, and so on and so forth. And it creates this parallel dependency pattern. And you see this is in contrast with the motifs like the data box where you have a block. So we hypothesize that actually the model has not memorized the sequence, the repeat, but rather has learned that it is a copy. So we have introduced repeats in artificial sequences of different lengths spaced by a different number of nucleotides. And as you see here, if you introduce a completely random repeat of 100 base pairs, 800 base pairs away, you can see the parallel pattern showing up. And indeed, if you start introducing repeats with bigger lengths, then these dependencies also increase, indicating that the copying operation has been captured by the model, and the model does not simply memorize the repeats. Interestingly, this is also valid not only for the copying operation, but for the reverse complement operation. So if you introduce a sequence and its reverse complement, the model is also going to link it in the dependencies. The dependencies allow us also to evaluate between different language models without any kind of fine-tuning. So you see again the example of the tRNA, and this is the ground truth secondary structure, and the dependencies from different DNA language models available out there. And you see, for example, for Rinalmo or species LM, you have the top dependencies belong to the arms here, the secondary structure of the tRNA. But you have models, for example, like EVO, which are autoregressive. So they predict the next token, which has an artifact where the first part of the matrix essentially doesn't have any dependencies. That is because autoregressive models only realize they are in a specific element once they have seen enough nucleotides. This can be mitigated by computing the forward and the reverse, but it is still worse than other bidirectional models. Models like nucleotide transformer, they have a chimerization which is non-overlapping sixmers. So they reconstruct non-overlapping sixmers. And this produces an artifact along the main diagonal, which tells you that the nucleotides inside the same sixmer artificially depend on each other. So this model can still have nucleotide pair specific dependencies outside of the main diagonal, but inside you can see these artifacts. Models like Hyena, you cannot really see any dependencies on these elements. So actually the major driver for the performance of this model on dependencies on the tRNA, in this case, is not the architecture of the model, but rather the data that it has been trained on. Specifically, models that have been trained only on human fail to see or to capture these dependencies between these conserved elements that are not very frequent in the genome. So now, since we are on the variant interpretation track, I'll finish with how you can use the dependencies for variant effect scoring. What we do is we take, rather than what it has been done, which is taking the ratio between the reconstruction of the alternative over the reconstruction of the reference, we take how does a variant affect the reconstruction of all other nucleotides in the sequence as a variant effect score, how influential that variant is on the reconstruction. And we first benchmark it on ClinVar, on non-coding variants, since we are using here a model that has been trained 2,000 base pairs before the start codon. It hasn't really seen any or many exon regions. And you see that broadly, this influence score is able to distinguish between benign and pathogenic variants. And if you compare actually the influence score with the reconstruction, so what I was telling you, the ratio between the reference and the alternative reconstruction, then the influence score is much better, but it does not beat some conservation-based metrics like phylop. And we think that it also has to do with the fact that there is some assortment bias here, because some variants in ClinVar are also supported by conservation. So we decided to go for a maybe more suitable data set, which is a promoter mutagenesis screen in human with 9 promoters. So here you see an example for the LDL receptor promoter used in this mutagenesis screen. And you have the nucleotide reconstruction probabilities from our species LM model. And you see that the reconstruction from the model is already aligning very well with the mutagenesis screen results. But if you take this reconstruction as a score to score the variant effects, it's still not better than taking alignment-based conservation scores like PhyloP or FastCons. But if you take the influence score, then this is much better than taking only the reconstruction, and even better than taking the alignment-based conservation scores. So the take-home here is that how influential a nucleotide is on all the others is perhaps a better predictor of its function than its own reconstructability. Okay, so to summarize, all of these observations have implications on variant interpretation. So now with the advent of language models, we can actually look at these dependencies and evaluate rather the influence of a nucleotide on reconstructing others, rather than a specific its own reconstructability or its own conservation. We also have implications on regulatory genomics, especially on less studied aspects of gene regulation, like post-transcriptional regulation, where we can now look or hypotheses in which elements might be motives. And also for a lot of non-model species where we don't have enough data out there. Also have implications in RNA structural biology for detecting functional RNA secondary structure and beyond. We also get not structured alternative structures, which I didn't show today. You also get tertiary structure. And in computational biology, specifically in how we design DNA language models and how we understand what they are really capturing. And last, but also I think very important, is that we have a new visualization tool. So several insights that we had across this project that prompted us for several systematic analysis came from first visualizing specific sequences. So now you have dependency maps which allow you to look at your sequence from a different angle. Okay, I would like to end with this, what I think is a beautiful example of a particularly crowded region in the mitochondria. And I would like to thank the amazing team working on this, especially Alex, which is co-first on this, Dani for the support on the structure, and especially to Julien for all the mentoring and the amazing opportunity to do exciting research. Thank you very much.
Questions?
Yes, questions.
It's so interesting. I'm wondering, you didn't say much though about your DNA large language model. I'm wondering,
yes,
is it different from some of the others out there?
So actually, we use more than
repeat the question.
Because people haven't.
Because you didn't speak in the mic, so maybe.
So
thank you.
yeah, no worries.
No worries.
I was asked about the DNA language model that we use. Actually, we use more than a dozen language models. Some of them are from our lab. Many others are not. So to make the point that it's actually quite broad. Some models have these different drawbacks, like I was explaining with this tRNA. But in the example, I'm interested on a specific example that I had shown. For example, for the variant effect, we are using a model that we have trained only on upstream, so 5' regions of genes in humans, so 2,000 base pairs, 5' of the start codon. And it is trained on metazoan species. Yeah.
Thank you.
Yeah.
Please, there's a microphone up front if you want to come up.
Out of all the models you tried, what model had the longest context length, and what was that context length?
Yes, so in theory, Evo can have a huge context length of in the maybe megabase because it's an autoregressive model. but we only tried it in shorter length sequences. But still, in this short range, this long range, these models that in theory can have a long range sequence do not even perform very well in terms of dependencies in the short range.
I couldn't fit more than like 512 in the EVO.
Sorry?
I couldn't fit more than like 512 in the EVO, but I think I had a small GPU.
Yeah, you can go. It depends on the GPUs you have, but nowadays these most models take until 12,000, so in the thousands range. That's what we have right now, mostly.
Go for it.
Yeah, I think Pedro. Okay, good. I'm wondering, I mean, I find this very intriguing because you have this one score, you just have the language model just learns all of these, patterns. Essentially, it learns transcription factor binding sites and is able to summarize this, right, without you explicitly telling.
Yeah.
But I'm wondering, in this particular example now, if you would now compute, right, using standard PWM of TFs, and then ask, like, okay, if I mutate it, what is sort of like the deviation, for TFs? How similar this is in the metric that you show on the right, how similar is this, right? Is it that you're just saying you're learning these binding sites and it's like a very nice way to, project this, easily with the score, or is there something in addition?
I think, well, it could be that the variant is hitting a motif site, and then maybe the PWM would already indicate something like that. But it could be that the variant is indicated and is hitting another site which influences the whole region. I mean, an example would be, for example, in the coding region, if you have a variant that induces a nonsense variant, then the likelihood of your whole sequence would change. So this is capturing more than just the motifs, but also other kinds of functional dependencies, I would say.
But why would the DNA model capture this? The implications you just made, wouldn't it just? Why? I mean,
why would it have captured this kind of dependencies? From looking at a huge amount of sequences where these nucleotides have been seen in specific contexts. So in the case of the coding sequence, the model has rarely seen any an in-frame stop codon on the wrong place. So things.
Yeah,
like
coding sequence I get. I'm just asking for the regulatory sequences. But I mean, okay, it could be combinations of those, and you destroy one,
Yes.
and then it impacts others. I see.
Yes, we see things like this, dependencies between motifs.
Okay, thanks.
So for all of your queries, are you just masking one position and then varying it based on another position, or are the queries more involved in that?
The query, we just mutate that position. We don't mask, we mutate that position, and then we check the probabilities of all models.
And then you look at dependencies all across the sequence when you mutate that one position?
You mutate that one position, and then with one forward from the model, you can get the probability per position and nucleotide across the whole sequence.
So you're generating forward from that position?
Yes.
Okay.
I'm just curious how we should be thinking about the relationship between this and like maybe some attention map kind of approaches. If you can
so
talk about that.
Maybe in some dependency maps, there will be some attention map which will have captured most of it. But what we see is that dependency maps, like you see splicing, for example, are actually composite of different layers of the model. So you cannot find the dependencies you see in splicing just in one attention map. They will be scattered across attention maps. Okay. So this is a more objective metric of like getting the information between these nucleotides. The attention maps would be a more indirect version.
Right. There wouldn't be an obvious way to sort of do a direct comparison of those two things.
Yeah, it is not trivial to aggregate all of these attention maps, and then some language models don't even have are not transformer based. Mm-hmm. So if you change and suddenly next year there is a new architecture, you cannot do this. Cool. So with this in silico mutagenesis is essentially a method you can do it for any language model or whatever model that predicts the probability for nucleotide.
Thank you.
Okay, I have a last one maybe, which I got asked by others. So what do you think if this is applied on protein language model?
Yeah, I think there is potential for this. Yeah.
What could we get?
we could get, maybe we'll be able to better score things like missense variants or variants which actually influence the structure as a whole and are not very local. I think we could better score this kind of variants.